Welcome everyone to this explainer. Okay, so did you know that a single tumor can harbor literally thousands of genetic mutations? But here's the catch. Only a tiny fraction of them are actually driving the cancer. If your goal is to really master clinical next-generation sequencing, or NGS reporting, whether we're talking cancer, germline, or clinical exome testing, you are in exactly the right place. Today, I'm going to be your guide as we completely deconstruct the anatomy of a clinical NGS report. We're taking the journey straight from raw, messy genetic data all the way to actionable patient care. Because, it's not just about memorizing technical bioinformatics terms, it's about mastering the clinical interpretation that actually drives real life-saving decisions.
So here is our roadmap for today. We'll start with a quick introduction to NGS reporting. Then we'll cover sample quality and metrics, identifying genetic mutations, and classifying variant significance. After that, we'll dive into biomarkers and clinical action, and finally, we'll look at building the final report. Let's just jump right in.
Section 1, Introduction to NGS Reporting, the Blueprint.
A clinical report isn't just some massive, intimidating spreadsheet of DNA letters. It's actually a highly structured document. It kicks off with the basics, patient information and sample details. Like, are we looking at a blood draw or a formalin-fixed tissue sample? From there, we move into the test methodology and the quality metrics, which basically validate the test itself. Then comes the real core of the report, identifying clinically significant variants and classifying them. Finally, we arrive at the absolute most critical part for the oncologist, which is the therapeutic and prognostic implications. Everything in this document flows toward one single goal, figuring out how to help the patient.
Section 2, Sample quality and metrics, the foundation.
So, the most brilliant bioinformatics analysis in the world? It means absolutely nothing if your physical sample quality is garbage. And that brings us to tumor purity. Tumor purity is simply the actual percentage of tumor cells present in your sample compared to healthy normal cells. Imagine you get a sample with a tumor purity of 70%. That's a great, strong sample. But if you have very low tumor purity, you've got a major problem on your hands. Why? Because the background noise from all those healthy cells can basically drown out the cancer cells, causing us to completely miss low-frequency genetic variants. Your foundation absolutely must be solid.
And that leads us right into VAF, or variant allele frequency. Think of this as the percentage of sequencing reads that actually contain the mutation we're looking for. Notice how a VAF of 100% usually points to a homozygous mutation or a loss of heterozygosity. A VAF around 50% often indicates a germline heterozygous mutation, meaning it was inherited, or maybe a clonal somatic mutation that's present in all the tumor cells. But I want you to consider the 5% or 1% range. These are low-frequency subclones. We're talking tiny fractions of the tumor. Without high tumor purity and really great sequencing depth, those 1% ultra-low frequency mutations, which, by the way, might be highly resistant to certain drugs, they would be completely invisible to us.
Section 3, Identifying genetic mutations, finding the errors. All right, our sample is good, so we zoom in to the DNA level to find the actual errors. The most fundamental split you've got to know in clinical genetics is germline versus somatic mutations. Let's make sure we are absolutely crystal clear here. Germline mutations are present from birth. They can be inherited, passed down through families, and because they're literally in every single cell of the body, you can find them in a standard blood draw. Somatic mutations, on the other hand, are acquired over time. They occur only in the tumor cells, they are not inherited, and generally speaking, you won't find them in the patient's normal, healthy cells. Now, once we're actually looking at the tumor, we have to separate the signal from the noise. That is where driver and passenger mutations come in. Driver mutations, like those in the EGFR or KRAS genes, well, they're the troublemakers. They actively contribute to the growth and progression of the cancer. Passenger mutations, though, they're just along for the ride. They're the backseat drivers of the tumor. They're present, sure, but they play no significant role in actually driving the disease. Our primary job in reporting is to hunt down those true drivers. So how exactly does DNA break? Well, in quite a few ways, actually. We've got gene fusions, where two totally separate genes abnormally join together, like your LK or ROS1 fusions. We look for copy number gains or losses, which is where a gene either multiplies itself or gets deleted altogether. There's loss of heterozygosity, or LOH, which is super crucial in BRCA-associated tumors. And then we have the structural code errors, splice site mutations that mess up how a protein is assembled, nonsense mutations that create a premature stop codon so the protein ends up incomplete, and frameshift or in-frame indels, insertions and deletions that alter the reading frame of the DNA itself.
Okay, so we've sequenced the DNA. The variant is found, the bioinformatics pipeline did its job flawlessly. But what happens next? A list of broken genes isn't a medical treatment, right? How do we take that raw biological finding and turn it into actual clinical meaning?
Section 4. Classifying variant significance, the danger scale.
We need to figure out if the genetic error we just found is actually what's making the patient sick. To do this, clinical labs universally rely on this 5-tier classification system from the ACMG. At the very top, we have pathogenic, meaning we have strong scientific evidence this definitely causes disease. Then there's likely pathogenic, which means we are over 90% confident it causes disease. Down at the bottom, we have likely benign and benign, which are just normal, everyday human genetic variations. So the really crucial point is right there in the middle, the VUS, or variant of uncertain significance. We found a mutation, sure, but the scientific literature doesn't definitively tell us if it's harmful or harmless. In everyday clinical practice, doctors generally do not change a patient's treatment based on a VUS. We simply need more data.
Now, while ACMG is heavily used for germline testing, when we're dealing with somatic variants in cancer, we often switch over to the AMP/ASCO/CAP tier system. It ranks variants specifically based on their clinical relevance. Tier 1 means the variant has strong, undeniable clinical significance. Tier 2 means potential significance, so maybe there's an investigational drug available in a clinical trial. Tier 3 is our unknown significance bucket, and Tier 4 is for those benign variants. This tiering system is great because it immediately tells the oncologist exactly how much weight to give the finding.
Section 5. Biomarkers and Clinical Action. Targeted Hope. This right here is the most impactful part of the entire report. Ultimately, what we're hunting for is an actionable variant. By definition, this is a specific mutation that can directly change a patient's treatment plan. This is the big eureka moment in a report. For instance, if we find a specific EGFR mutation, we know we can target it directly with an EGFR inhibitor drug. Or if we find a BRAF V600E mutation, guess what? There's a BRAF inhibitor for that. It literally transforms cancer from a generic, broad disease into a specifically targetable molecular puzzle.
To find that targeted hope, we have to analyze a few key clinical markers. We're looking at predictive markers, which tell us if a patient is likely to respond to a specific drug. We look at prognostic markers, like a TP53 mutation, which might unfortunately indicate a poorer future disease outlook. We also measure broad genomic signatures, like TMB, or tumor mutational burden, which is literally just the number of mutations per megabase. We ask, is the tumor's DNA repair completely broken? High TMB and microsatellite instability, or MSI high, are massive red flags for the cell itself. But clinically, this is actually amazing news for the patient because it strongly points them toward life-saving immunotherapies. Lastly, some of these tests are companion diagnostics, meaning the FDA legally requires this exact NGS test to be run before a doctor can prescribe a specific drug.
Of course, when we recommend these targeted treatments, we absolutely have to back it up with hard evidence. That's where OncoKB evidence levels come into play. We use them to prove to the clinical team exactly how strong the scientific backing really is. Level 1 is the gold standard, an FDA-approved biomarker for that exact cancer type. Level 2 is standard of care evidence. Level 3 means strong clinical evidence exists, but maybe it's a bit limited, or it's an off-label use. And Level 4 is just biological or preclinical evidence, meaning, hey, it worked in a petri dish or an animal model, but it is certainly not ready for clinical guarantees.
Section 6. Building the final report. Putting it all together.
Let's look at how bioinformaticians and variant scientists actually stitch all these pieces together into the final document that goes to the oncologist. If you're working on solid tumor panels, there is a core cast of heavy hitter genes you simply must know. EGFR, KRAS, BRAF, TP53, PIK3CA, HER2, the fusion genes like ALK, ROS1, and RET, and of course BRCA1 and BRCA2. Mastering the specific hotspot variants, the gene fusions, and the copy number changes of these specific genes is absolutely non-negotiable for clinical reporting. These are the genes driving the most common cancers, and frankly, they are the ones with the most targeted therapies available today. And to interpret these genes, we definitely don't just guess. We cross-reference every single thing we found in the patient sample against massive, globally curated clinical databases.
We use ClinVar to check for germline pathogenesis. We use COSMIC, the catalog of somatic mutations in cancer, to see if the tumor mutation has been reported before. We check NOMAD to ensure the variant isn't just some common, completely harmless population trait. And we use OncoKB and CIVIC to gather the absolute latest therapeutic evidence.
When it's finally time to write all of this down in the report, we have to make sure scientists and doctors all over the globe are speaking the exact same language. For this, we use standardized HGVS nomenclature. For example, if you see C.35G to A, the C tells you we're looking at the coding DNA level, at position 35, where a guanine was abnormally replaced by an adenine. If you see P.G12D, the P means we're looking at the protein level, where the amino acid glycine at position 12 was swapped out for an aspartic acid. It is a highly precise, universal coordinate system for genetic errors.
And that, everyone, is the true anatomy of a clinical NGS report. Mastering these terms isn't just about crunching data or memorizing a bunch of databases. It's about bridging that massive gap between a raw genetic sequence and a real life-saving clinical decision. Because behind every single VAF percentage, behind every actionable variant, and behind every tier classification, there is a very real patient waiting for a treatment plan. If we have the power today to turn a sequencer's raw output into targeted hope, just imagine what diseases we might be able to cure tomorrow as these genomic panels become even more advanced. Are we on the verge of making cancer a completely manageable condition? Something to think about. Thanks so much for joining me on this explainer, and keep digging into the data.